Welcome. Today we are diving deep into the very tissue that makes you, well, you. We're going to explore how the nervous system is built for incredible speed, how it processes information, and how it's protected by this amazing fortress of specialized cells. Getting a handle on this histology is absolutely key to understanding everything from a simple reflex to our most complex thoughts.
So let's just break it down with a simple example. Imagine you see a baseball flying at you. That's the sensory input. Your brain and spinal cord then kick into gear, performing what we call integration. They're instantly calculating the ball's path and figuring out what to do. The result, a motor output, which is a signal sent to, say, the muscles in your arm telling you to catch it. That whole lightning-fast circuit is powered by nervous tissue. All right, here's our game plan for today. We'll start with the big picture, the nervous system's blueprint. Then we'll meet the cells doing all the work. After that, we'll look at the need for speed and what happens when things get damaged. And finally, we'll explore the incredible fortress that protects our central nervous system. Okay, let's jump right in. Section 1, the nervous system blueprint. Let's start with how this whole thing is organized. So, our nervous system has two main parts. Think of the central nervous system, the CNS, that's the brain and spinal cord, as the headquarters. It's where all the big decisions are made. The peripheral nervous system, or PNS, is everything else. It's this huge network of nerves that are like the communication lines, carrying messages back and forth from headquarters to the rest of the body. Section 2, the cellular workforce. Now, let's zoom in and meet the building blocks. The entire nervous system is really made of just two main types of cells. So you've got two major players here. On one hand, you have the neurons. They're the stars of the show, the communicators. They're excitable, which means they can generate and send electrical signals. But then you have the neuroglia, or glial cells. These guys are the unsung heroes. They aren't excitable, but they are absolutely essential for supporting, protecting, and insulating the neurons so they can do their job properly. First step in our glial cell lineup are the astrocytes. I mean, look at them. They're star-shaped. They're the most common glial cells in the CNS. And you can see right here how they physically connect neurons to their blood supply. The capillaries. They're literally the bridge for nutrients to get across. But being a bridge is just the start. Astrocytes are master regulators. They're not just supplying nutrients, they're also maintaining the super delicate balance of ions like potassium and calcium. They clean up leftover chemical messengers called neurotransmitters, and, this is a big one, they help form the blood-brain barrier, which we're definitely going to talk more about later. Then we've got the specialists teams. Microglia are the resident immune cells of the CNS. You can think of them as tiny security guards or janitors, constantly patrolling and gobbling up any debris or invaders they find. And then you have ependymal cells. They line the fluid-filled spaces in the brain and spinal cord, and their job is to produce and circulate that protective cushion of fluid, the CSF. Now this is a really cool comparison, showing a key difference between the CNS and the PNS. Both systems have cells that create insulation, but they go about it in totally different ways. See on the left, in the CNS, a single oligodendrocyte reaches out and wraps multiple axons at once. But in the PNS, it takes a whole team of Schwann cells, with each cell dedicating itself to wrapping just one little segment of a single axon. Section 3, need for speed. Okay, so that insulation we just saw, it's called myelination, and it's the secret to creating a biological superhighway for nerve signals. Let's see how it works. So, what exactly is this myelin sheath? Honestly, the best analogy is the plastic insulation on an electrical wire. It's this fatty coating that does three critical things. It protects the axon, it insulates it so signals don't get crossed, and most importantly, it gives a massive, massive boost to the speed of the signal. So how does this wrapping actually happen? Well, in the PNS, a Schwann cell basically grabs onto a piece of the axon. Then it starts to rotate, wrapping its own membrane around and around and around, kind of like rolling up a sleeping bag really tightly. And what you're left with is this thick, multi-layered insulating sheath. And this is where the design gets really brilliant. See, the insulation isn't one solid piece. There are these tiny, exposed gaps called the nodes of Ranvier. The electrical signal doesn't have to travel down the whole wire. Instead, it literally jumps from one node to the next. This process is called saltatory conduction, and it is way, way faster and more energy efficient. It's an incredible biological shortcut. So what happens when this complex, high-speed wiring gets damaged?
The answer here has huge clinical importance for everything from a sports injury to serious neurodegenerative diseases. When an axon gets cut or crushed, the part that's been disconnected from the main cell body, it starts to die. You can see it on the right. The myelin sheath begins to break down, and the axon itself just degenerates. It's a process known as Wallerian degeneration. Which brings us to the million-dollar question, right? Can a neuron actually regenerate? Can it fix this kind of damage and reconnect its circuits? Well, the answer, fascinatingly, is it depends.
And it depends entirely on where that damage happened. Now, this is a huge takeaway point. In the PNS, there's actually a pretty good chance of regeneration. The Schwann cells form a little tube to guide the regrowth, and they create a super supportive environment. But in the CNS, it's a completely different story. The environment there actually works against regrowth, which is a major reason why recovering from things like spinal cord injuries is so incredibly difficult.
Okay, for our last section, let's zoom back out and look at the amazing protective structures that stand guard over the brain and spinal cord. The CNS isn't just rattling around inside our bones. No way. It's carefully wrapped in 3 distinct layers of tissue, called the meninges. These layers provide a stable, protected space for our most vital organs. So working from the outside in, first you have the dura mater. It's Latin for tough mother, and it really is. It's a leathery, strong layer. Below that is the arachnoid mater, which looks kind of like a spider web. The space under it is filled with that cerebral spinal fluid we talked about, which acts like a liquid shock absorber. And finally, clinging to every single fold and groove of the brain is the very delicate pia mater. The final, and maybe the most incredible layer of security, is microscopic. It's the blood-brain barrier, or the BBB. And this is not just a simple wall. Think of it as the world's most exclusive nightclub bouncer. It is a dynamic, highly selective border that decides exactly what gets to pass from the blood into the brain's pristine environment. The way this thing is built is just remarkable. It's not one thing, but whole team effort. You've got the cells lining the blood vessels, but they're sealed together with what are called tight junctions. Think of it like they're superglued. Nothing gets through. There's a thick membrane, other cells called pericytes, and then, remember our friends the astrocytes? Their end feet wrap around the entire vessel, creating that final seal. This barrier is so good at its job, it actually makes it a real challenge for doctors to get medicine into the brain. So, to wrap it all up, from the grand organization of the CNS and PNS, all the way down to the specific jobs of each glial cell, the incredible engineering for high-speed signals, and its amazing multi-layered defenses, you really have to conclude that the nervous system is an absolute masterpiece of biological engineering.